Good afternoon. Welcome, everyone, to today's conference call. At this time, your lines have been placed on listen-only for today's conference until the question-and-answer portion of our call, at which time you will be prompted to press star 1 on your touch-tone phone. Please ensure that your line is unmuted, and please record your name when prompted so that I may introduce you to ask your question. Our conference is being recorded, and if you have any objections, you may disconnect at this time. I will now turn the conference over to our host, Ms. Irene Iyer. Ma'am, you may proceed.
Thank you. Hello, I am Irene Iyer of CDRH's Office of Communication and Education. Welcome to the FDA's 28th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Toby Lowe, Associate Director of the Office of In Vitro Diagnostic and Radiological Health, and Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene. Thanks, everyone, for joining us today. My update today is that we updated the website that we have for the reference panel comparative data. So this is the reference panel that we've been sending out to test developers to characterize their LOD in comparison to other tests. And we first published data a few weeks ago, and we just published some new data that's come in since that the first batch. So I believe we added somewhere around 20 to 25 new tests to the data that we posted there. And now I'll turn it over to Tim for his update.
Thank you, Toby, and thanks to all for joining us again today. First thing I wanted to do is to give a shout out to those who are critical and so important for us to put on these weekly calls. First of all, you're very familiar with Irene, but her backup is Kemba. And folks that get involved with transcribing this and posting that. Of course, Toby and the operators and everyone else. There's a little bit of an echo, but I apologize for that.
Oh, now you're better, Tim.
Clear up. Okay,
yep,
good.
That fixed it.
Thanks. So thanks, Irene, Vicki, Kemba, everyone. Okay. I wanted to speak about the quality of EUA submission. So it's important to have a complete, well-organized submission, including your validation protocol, so we know that how your validations were performed and whether or not those were accurately and correctly done. Those are all important for us to move swiftly towards an authorization decision. You know, our expert reviewers have expressed to me a growing frequency of getting bogged down with less than optimal submissions. And they have spent a valuable time on working with these developers and trying to help. And in the process, of course, has delayed review of other applications. We're in a new phase now, and we've authorized quite a few tests, and therefore we are going to take steps to correct this situation so that we can work through all submissions as quickly as possible. So please take steps to have the best quality submissions. If you've already submitted and you want to update and make updates to it, please do. And then when we have questions, please address them directly and clearly and as quickly as and completely as possible so that we can work through those submissions with you and then move on to the others as quickly as possible. So I thank all in advance for the help with this. And so with that, we'll turn it over to question and answers. Hello?
Your line is
Tim,
open.
are you there?
Yes.
We can hear you.
All right. Yeah, moving to Q&A now, if possible.
At this time, if you would like to ask a question, please press star 1 on your touch-tone phone. Please ensure that your line is unmuted, and please record your name when prompted so that I may introduce you to ask your question. Please stand by for questions. Our first question is from Savannah Esteve. Your line is open.
Hello, this is Savannah Esteve with UserWise Consulting. I have a question about EMC compliance. So in the molecular diagnostic template for commercial manufacturers, underneath the software validation section, the FDA recommends that manufacturers perform EMC testing to comply with IEC 60601-1-2. We have a client who is tested for EMC through compliance to the IEC 61010 standards. Is it possible that the FDA would accept the IEC 61010 standard compliance instead of the IEC 60601 standards?
I would want to verify the standard that the entire center is moving to. I think it's the first one you mentioned that's in our templates. You can double check at the templates email address. But we are making that transition and understand that there's some opportunities to grow through that transition. So we are flexible on dealing with potential deviations from what we'd like to adhere to and are open to alternate mitigation. So, and that can include labeling. But when you get into it's important on a one-by-one basis to understand that at a granular level. But obviously we want, if there's instruments, electrical instruments used, we want to make sure that no harm comes to users of those instruments.
Okay, that makes sense. Thank you.
All right, thank you.
Our next question is from Shannon Clark. Your line is open.
Hello, Shannon Clark, also with UserWise Consulting. There isn't currently a clear template that outlines home use testing for antibody lateral flow test kits. Should we just go ahead and follow the antigen home use instructions with testing of 100 users who are negative for COVID-19 in order to obtain an OTC designation, or 30 lay users who are negative for COVID-19 if we're pursuing a prescription-only indication?
Well, that's certainly a good start. We're supportive of that. We are working on a template specifically for that. We have enough built up thoughts about that to be able to provide via the template's email address with some recommendations. So if you reach out to the template's email address and ask for the home testing serology recommendations, then one of our team members can assist you with the best possible and most up-to-date recommendations that we have.
Great. And do you have any concerns about testing only COVID-negative individuals and displaying positive results to those individuals in order to test that scenario?
Overall, we want to ensure that in the non-lab setting, that accurate results are obtained, accurate readings are obtained, and accurate interpretations, especially in the OTC, over-the-counter, where a clinician is not involved in the testing for those individuals. So I don't remember the details of the current recommendations for serology at home, and therefore, do request those recommendations, and then if you have follow-up questions, those contacts can work through any potential alternatives.
Okay, thank you.
Thank you. Our next question is from Luis Perlmutter. Your line is open.
Yes. Anyway, Tim and Toba, thanks for taking my call. Besides the Biomax now from Abbott, is there any other rapid antigen tests been approved?
Yes. We've authorized 4, I believe. The Binax, to my knowledge, is the only one authorized to date that doesn't require an instrument. And certainly there are more in development that won't require an instrument. Instruments in the area of rapid testing have been deployed for other respiratory pathogens such as flu and RSV, and there can be obvious benefits in using an instrument, but in the current pandemic, and prior to this pandemic, we also authorized non-instrumented rapids and continue to do so.
Yeah, because I was thinking of the non-instrument tests, but just to go on, has anything been approved for saliva-based rapid antigen tests?
Not yet. In our conversations with experts in the field, there has been some concern about the use of saliva for rapids and the sensitivity, and given the fact that there's not an amplification.
Yeah, because it would be nice to convert. Yeah, but I understand the concerns. And of course,
we're at the end of the day, data supporting use of a specific sample type will be reviewed. We don't make any preconceived decisions about this.
Right.
We're obviously into uncharted territory, looking at saliva as a sample type for respiratory viruses, but we've authorized it for molecular tests. And while I can't encourage it for antigen tests specifically, I'm not dissuading developers either. If you have ideas, come to us. If you have data, come to us.
Great. Okay, thanks for your answers.
Once again, if you would like to ask a question, please press star 1 on your touch-tone phone and record your name. We have a question from Franco Calderon. Your line is open.
Thank you very much, Dr. Stencil. I have a question regarding LOD for antigen tests, and if you could also speak a little more about sensitivity and specificity. I know regarding the LOD, you've said a couple of times before that the focus should be on the overall clinical performance because there are no universal standards set for LOD. I would appreciate it.
Yeah, so, I think our expectations both for those that are prescribed tests for antigen and as well as those that are OTC, and in the home environment. We've made, our recommendations for performance, at least for sensitivity or PPA, quite clear. That we would like to see at least 80% sensitivity for a single result compared to a high sensitivity molecular test, at least during the period of time when an individual is considered to be infectious. So that period is when we typically think, after infection and when they begin to shed virus, in perhaps the first 5 to 7 days of shedding, and at least that's the period that I correlate with symptoms for those patients who do have symptoms. We would like to see that, overall that at least 80% sensitivity for that period. That does mean that potentially during that period your test is missing 20% of people who could be infectious. And then, certainly we've heard arguments made in the public arena for authorizing much lower sensitivity tests, significantly lower, significantly below 50%, and as low as 30% or even lower. And, if a test then misses more than 50% of the people who could be infective, we would have serious concerns about that kind of test. Now, we are open, as Dr. Shuren and I have expressed in a recent op-ed in The Hill, to using serial testing. It's very possible that a test that individually might have a performance below our recommendation, say 70% PPA, that if used in serial testing, perhaps a two-pack, two tests in a pack, and would rely on testing twice, the same individual twice over a set period of time, maybe day 1, day 2, day 1, day 3, something like that. And we've expressed, very much flexibility to say we can look at the combined performance of such a strategy. And if it does do an excellent job of hitting our, our minimum recommendations, that we can consider that for authorization. However, we do think that the vast majority of people who are infective or should be infective, that is, they have the ability to infect others, we would want to see identify those individuals.
Okay, thank you. Thank you for that. If I may add a couple of related questions. So can you speak a little bit to the LOD? I know in the past 2-3 calls you've said that LOD should not be such a big deal in antigen tests, rather the overall clinical performance. And then the last one is perhaps not something that you do, but my test was developed and validated in China. Okay, so we have data for that. However, the biggest hurdle I'm having is finding a CLIA lab, whether it is private or public or an academic lab, to conduct the U.S. validation, both analytical and clinical. Does FDA provide any resources, recommendations for someone like me that's trying to do this? I have the test. I can produce 300,000 of these a day, but I do have that hurdle of the validation, not being able to, after multiple contacts to, again, public, private, academic institutions, and everybody is extremely busy.
Yeah, we have heard that there are some contract research organizations out there that have been very helpful to developers, and we don't give out recommendations for her to use, but certainly that's a pool of folks who can potentially help. I think what you're probably talking about is doing the point-of-care studies to demonstrate that non-laboratory users in the point-of-care setting can perform the test accurately. And while we would love to see that work done in the United States, that's not a formal requirement. It is a requirement, though, to do that testing in English and perhaps even in Spanish. Look to the template because that's the U.S. target population. So, so hopefully that's helpful.
Could you please clarify? So are you? Am I misunderstanding when you say that the performance data that we already have for the test, if it's presented in English and or Spanish, could be acceptable, or is that?
If you've done the point of care study, that is not in a laboratory, not in a laboratory with formally laboratory trained people, but you've done it in a clear-waved environment with non-laboratory healthcare professionals. So it's all in how the study was carried out in the target population, because when we deem a test as clear-waved, we expect that test will then be performed by non-laboratory users. Even these days, by, say, officials at schools and businesses who have never, or even nursing homes who have never done this kind of testing before, and they have newly acquired a certificate of waiver from CMS CLIA. And then finally, and I want to wrap up these questions because I have a feeling there's more questions from callers, is LOD. Probably many outside the FDA have made more a deal of LOD than we do. First of all, if there's not an easy way to harmonize how LOD's experiments are performed, it's very hard for us to assess individual LOD determinations. We do require it. We expect people to do it, but it is a lesser review item than for actual clinical study performance. That is, what is your sensitivity and what is your specificity? And then if you are, want to do things like home testing or point of care testing, that you've done the appropriate studies to show accuracy in those settings as well. But thank you for your question, the opportunity to address some of those.
Thank you.
We'll need to move on.
Thank you. The next question comes from Sharik Koshik. Your line is open.
Hey, Tim and Toby, thank you for taking these calls, taking the town hall every week and taking our questions. Tim, it's basically I wanted to ask a question about the issue of the quality of submission that you raised. Could you elaborate on when you said you needed validation protocols? Did you guys actually want to see the protocol before it's implemented? Or, I mean, the way I've been doing my submissions is to actually give a clinical trial report or an analytical validation report that briefly describes the protocol. Could you give us some examples of what you want us to give you for making the submissions better?
Yeah, in a perfect world, where you're doing pre-subs or Q-subs, or in the case of EUAs, a pre-EUA, we have a lot of time, which we don't. We don't have the time or resources to give as much feedback as we would love, which is why we provide templates that provide hopefully very clear recommendations on study designs and expectations. When we receive the data, if we just receive the data and we don't receive information about how the studies were carried out, it's sometimes hard for us to assess, whether this directly applies to what we review. So if your summary of how you did this is clear enough and our team doesn't have questions, then we're fine. We just want to very clearly understand, what were the requirements you set on the study, what are the inclusion, exclusion criteria, the settings, the type of users, all that is very important for us to understanding the performance and being able to make decisions as quickly as possible. Hopefully that clears things up.
Yes, it does. Thank you very much.
question comes from Tom Slovic. Your line is open.
Yes, I would like some advice on what is the most appropriate product code to use for a saliva collection kit with VTM and also a swab collection kit with VTM. I've seen an amazing range of different codes used.
I'm not going to be able to provide you with those codes, but we have experts on the team who can. I don't know if Toby has them at hand. Important, yes.
I don't know off the top of my head, but if you email the mailbox, we can get you some answers on that.
Thank you very much.
Mm-hmm.
Thank you. The next question comes from Annie Bell. Your line is open.
Hi, thanks. Two questions related to the serology template for manufacturers. Are there any limitations on using archived samples for a semi-quantitative serology test?
We are working on our recommendations for semi-quant neutralizing antibody tests. I think our thinking is advanced enough that if you ask that question into our templates email box, we can provide a response, and without double checking with the team on what they would like. I'll just say generally we try to be as flexible as possible with archived samples and in order to speed development and speed access to technologies.
Okay, great. And then one other question on the same template. Does there have to be more than one lot used in the robustness studies? Or can one lot of reagent be validated?
It depends. By and large, we've been not asking for that for EUAs. There are specific examples, though, in situations where we may.
Can you give an example where you might ask for that?
Yeah, I would say that it's uncommon, but it happens, and I would actually, I think it's best to handle that on a case-by-case basis. If you have any concerns about anything that's asked and you think it's not least burdensome, you can try to get a hold of me through the templates email and you can give me some specifics.
Sure. And specifically for stability, would more than one lot be required?
That is a great question. I don't know the answer to that off the top of my head for EUAs.
Okay.
And I would defer to the templates and our team for that. In general, I don't believe so, but I'd want to have you double check with our teams.
Okay. Thank you for your help.
Thank you. The next question comes from Sean Gemmel. Your line is open.
Good morning, Dr. Stencil. Good morning, Toby. Thank you for taking the call. I represent a high-complexity accredited laboratory, single laboratory located in Carlsbad, California. We submitted our EUA for a high-throughput 20,000 specimens per day PCR assay. Our EUA has been under review for a little bit over 3.5 months. To your question, to your comment earlier, we did address questions from the agency on software validation, provided that additional information. And we've answered all of FDA's questions pertaining to our EUA application. At this time, we're still awaiting an EUA approval or response from FDA for any outstanding items. Understanding that our application has been under review for 3.5 months, we're confident we've answered all questions. Can they provide an estimate of when an EUA approval should be anticipated? if our organization is confident that we've addressed all of the agency's questions in our EUA application.
Yeah, so we don't address specific applications on the open call. If I haven't already been approached by you or a representative of a developer, I often get these emails and then do look into them, and as soon as I can get back, I do get back. But if I haven't been brought in to your knowledge, you can send an email to the template's email address, and I can look into the specifics of the situation. And, of course, we do allow, via the guidance, for those that require an EUA, but where there's allowance for launch of that test prior to authorization through the guidance. We allow that pathway for a very specific reason, because we are dealing with volumes of submissions, and we didn't want to delay access.
Yeah, Dr. Stencil, thank you. Our company is listed on the FDA website as a high-complexity CLIA laboratory offering testing. However, it states it is currently not EUA authorized. And so I know the question has been asked before. Callers, we understand the great number of applications, but on behalf of the organization, we were just looking for an estimate of when we should anticipate our final response from the agency. So if we haven't...
If you contact me with the name of the submission by somebody who has authority to communicate with us, then I will, and through the template's email address, I ask for Tim Stenzel, and they'll get that over to me, and we'll look into it, and we'll provide a specific answer.
Great, Dr. Stenzel. Yes, I'm the point of contact on our EUA application, and I will forward an email to you through the EUA templates address. And what's taking the question?
Thank you.
Our next question comes from Jeff Gray. Your line is open.
Hi, this is Jeff Gray from Groltex. Thanks again very much for having these meetings. Very helpful. Question, over the last couple of meetings, you've said that you still had not had any submissions for an at-home POC virus test. Is that still true? And then the question is, why do you think that is? I guess I'm looking for your opinion there.
That's still true to my knowledge. We've had a home non-lab test template out there for a while. We certainly engaged with a number of parties. I've certainly personally encouraged a number of developers to advance their programs. I don't know. Honestly, I don't know.
I'm just looking to learn here. Do you think it's a market reason or a regulatory reason or a reimbursement reason?
Well, something goes OTC. I don't believe there's reimbursement. It's patient out-of-pocket sort of testing, or it's whatever entity is doing the testing out of their pocket, to my knowledge. But I would refer you to CMS with regard to any specifics about reimbursement for that.
Okay.
If something's by prescription, typically it is covered. We clearly allow prescription tests to be performed in the home. We've authorized those for other non-COVID tests for home use by prescription. And anyways, we're very open to it. We're very open to working with developers. We've provided, recommendations for validations. So we're open for business.
Okay, thanks. Sorry, one other question, kind of a tactical one. If one submits a pre-EUA submission and gets on a kind of a EUA track, if you will, and then if largely the EUA program goes away or something changes, are submissions already in the queue still going to be approached with EUA, or how do you see that?
I'm sorry, can you ask that question again?
Sure. If you make a submission under an assumption that you're going for an EUA and you're partway through that track, if then the EUA program goes away, will you phase it out and still approach people who already have submissions under EUA, still consider them for EUA, or if it goes away, it's just done, that's it? Whoever's in the queue doesn't matter.
Yeah, that's a real excellent question, and I don't have an answer. I'm not sure if Toby does either. I mean, certainly, we have a number of open emergency authorizations from prior emergencies still, and we have not closed those down from the agency perspective. And we would support a very amenable transition plan so that those that have developed tests that could, help out here. So, for those that have already been authorized, there's the question for them. Specifically, I don't have a ready answer for those that have submitted but haven't been authorized, except to say that it's, from our perspective, we want to have a smooth transition, and we want to give all developers who want to make that transition in general sufficient time to convert.
Okay. Thanks very much again for having these. Appreciate it.
Yep.
Our next question is from Patrice Milo. Your line is open.
Thank you very much. This is Patrice Milo from Pine Trees Health, and I'm interested in your perspective relative to surveillance testing, and what is your guidance on point-of-care molecular tests being used for currently unmet surveillance testing needs in settings such as schools? And in conducting that, how should results be properly reported and acted upon? And how should our tests be labeled as such, for example, for research use only?
Yeah, we've gotten some inquiries about that. Toby, are you up to responding to this question?
Sure. So I think, it's important to take a look at our website where we discuss surveillance testing and what surveillance testing is. So you'll want to make sure that, that your the intent of your testing is truly surveillance and not screening. And then we have indicated on our website that FDA does not actually regulate surveillance testing if it truly is surveillance testing. So then we would recommend that you also take a look at the resources that CMS has put out about surveillance testing and about ensuring that you have the appropriate CLIA certification as necessary for the testing that you're doing. And then I believe that CDC has also put out some recommendations regarding reporting and calling out the differences between surveillance and screening and diagnostic testing.
Great. Thank you very much.
Sure.
Our next question comes from Kadu Moody Venkat. Your line is open.
Hello. Good afternoon. Can you hear me?
Yes.
Yeah, thank you for taking my call. I greatly appreciate the town hall and your clarifications. Earlier, you mentioned about the quality of the submissions. I have a question regarding that. When the review is being done currently, does it mean that the earlier submissions have gone through a linear review and now the submissions under review are being reviewed with more rigor? Is that way, or it is the same format it is being done, and because of the time, if the submission quality is better, the review can go faster? Can you just clarify, please? In general, our review process hasn't changed. It's just earlier on, when there were fewer submissions, we were trying to be as helpful as possible and perhaps taking more time
than we can afford now on a given submission. So, and sometimes we will ask for information, and there's some dialogue about the necessity of getting that information, and because of the volume and because of our desire to work through all of the submissions as quickly as possible, we want to shorten those conversations, make them as brief as possible, and asking developers to help us get the information that we need to make as quick a decision as possible.
I mean, does that mean that the people who submitted earlier because of the early mover advantage, they got away with less stringency compared to the bar is high now for review?
No, not at all. Our expectations or recommendations as far as performance goes has not changed. What I'm saying is that if a submission has issues and they're not quickly addressed, we may triage those to a lower priority while we move on to the next. So we would encourage developers, when we ask a question, to be succinct, quick, clear, and provide us the information, because otherwise we're going to have to move on to the next submission, in all fairness to other developers.
Thank you. Greatly appreciate your comments.
Our next question is from Gustavo Higre. Your line is open.
Hello, my name is Gustavo Higre. Can you hear me?
Yes, we can.
Oh, well, good afternoon to everybody, and I want to thank you for this town hall meeting. I think it's a great idea. I'm looking forward to hearing some of the previous ones to be a little bit better informed. My question is regarding a real-time PCR machine. It's made by a Chinese company called Hongxi Shanghai, and the model is a Plan 96P. It appears on an EUA ISU of a Korean company. And I would like to know if there's a way to use that machine for other tests that have an EUA.
So we're open to whatever instrument a developer wants to use, as long as it works. And if we've already authorized an instrument, obviously we've had a chance to try and understand its performance, at least with regard to the bars that we've set for EUA applications, which are much, much lower than our standard applications. And we do waive significant quality system requirements to make that, to allow for that to happen. So if you see something that has been utilized already in an EUA authorization, it's, you can certainly ask if there's any issues with that of our staff, but in all likelihood, we wouldn't have authorized that unless we had sufficient confidence in both the test and the platform it's run on. So, but we don't negotiate, terms or provide typically intermediary services. So feel free to reach out to any other developer of any technology in our space that you feel would be beneficial to your own program.
Okay, so I understood that if the equipment was authorized for test XYZ, it can be used and validated for another test that also has an EUA?
We wouldn't prohibit it, and as I said, if it performed once already with one developer, it's likely to perform again for anybody else. It's not a requirement, though. I mean, it's not a requirement for developers to use only instruments that we've for which we've allowed authorization for in developer submissions. You don't have to use something that's been previously authorized. You can use whatever you want as long as it performs well and meets our expectations.
And my next question is regarding the expansion region. The EUA says to use a QIAGEN expansion region, and we would like to know if it's possible to use another expansion region that is EUA approved.
Yeah, that's a question that I'm just not aware of that I would understand enough at this point. It sounds very specific. So for those kind of specific questions, I think it's best to go to our templates email address. But again, we're open to whatever technology that works, whatever extraction technology that works. It doesn't have to be previously authorized, as long as it works.
Great. Well, thanks a lot. That answers my questions.
You're welcome.
Our next question is from Dwayne Porman. Your line is open.
Hi. Thank you so much for holding these town halls. As others have stated, it's been very, very helpful. My question is in relation to the process for the notification pathway for commercial manufacturers of serological tests. After a manufacturer has sent in the notification to the template email address, could you just go over the process that occurs prior to the test being listed on the FAQ page and what the usual time frame is for that process? And I know time frames are really difficult for you to speak to. But if you could just give a little more information about that process, I'd really appreciate it.
Yeah. So if something comes in into our email templates box or wherever the particular box it comes into, we do a quick vetting of that request, and then usually within 2 business days, you should receive information back and confirmation of your request, and hopefully, if there's no issues, confirmation that your notification has been received and accepted, and then it goes to the web service folks who post that as soon as they can. So if there's a delay beyond 2 business days and you receiving word back about whether your notification request was allowed, then I would ping them again, email them again, and ask for an update as soon as possible.
Good. Okay. Thank you very much. That's very helpful.
Uh-huh.
Our next question is from Lubna Syed. Your line is open.
Hi, Tim. Can you guys hear me okay?
Yes.
Great. Thanks for taking the call. I know we're running out of time here, so I'll just get right to the point. I had a question about the website where you list the IVD authorized tests. It seems for recent approvals, you have to expand the date EUA first issued in order to get at the detailed information where previously it was at a top level, and that's great. My question is more around the authorized setting listing. For one of the recent authorizations on September 24th for a Cepheid that's on the public database, I'm not asking for anything private, it seems like there's a point-of-care IFU as well as a lab test IFU, but the authorized setting listing is only listing the H and not the W. Previously, when I saw authorizations for point-of-care that also had lab tests, there was an authorized setting listing with HMW, and I'm wondering if this is a new nomenclature or if we're still going to be able to sort on HMW in terms of authorized setting. Perhaps this was a glitch. I don't know, or something changed.
I would double check. Try refreshing your page and maybe clearing your cache, because I'm looking at it right now and it does show HMW for me.
Okay.
Entry.
Okay, perfect.
Yeah, I've just double-checked that in both for their SARS test and for their panel test, including flu and RSV. It does list all 3 designations.
Okay, I was looking specifically at the September 24th authorization. That's great. If that's fixed, that's wonderful. And it's just a glitch that was on my computer. So thank you for clearing that up.
Uh-huh.
Sure.
Our next question is from Ashani Fernando. Your line is open.
Actually, my question got answered during the previous clarification, so thank you.
Thank you, ma'am. We move on to Laura D'Angelo. Your line is open.
Thank you, and thank you guys, obviously, for all the work that you have been doing. It's really appreciated. I have two questions. I'll be very quick. Can you elaborate just briefly on the transition you mentioned, or is this, is this an impending regulatory transition, or is it just kind of like the passage of time and a little more attention to detail? I know that we are expecting some new templates and some updates, including, antibody lateral flow home use. But is this transition kind of a regulatory thing, or is it kind of more of an attitude? Is there going to be a formal announcement?
So we are working on a guidance that obviously has to be cleared within the U.S. government before it's posted on a transition path. And as soon as that can be finalized and posted, I can't. I'm not in control of when that gets posted. But we certainly anticipate the question, and we have for a while, and the potential concern. We don't want to over-minimize the concern, but it is, as I've stated and others have stated, our desire to have a very reasonable transition period from EUA authorizations to full authorizations.
Okay, so when you said earlier that we're in kind of a new phase now, that was just kind of a general thought?
That had to do with, our ability to spend as much individual time with the developer as we may have had in the past or taken in the past. It had nothing to do with the transition and everything to do with the fact that as the number of submissions are growing, our desire to get through them as fast as possible. And now that we have a lot more templates and recommendations for validations and very clear recommendations and expectations, we think that due to that work, that our conversations with individual developers can be much more efficient, and that will help us speed through the applications.
Okay, that makes sense. And then just super quick, and I understand if you can't say anything kind of too strongly about it, but just status update on international LOD standards. That's still kind of in the hopper with PCR coming first, followed by antibody and antigen.
They're all being worked on. We're not the main drivers. The FDA is not the main drivers, but we're at the table. My understanding is those are still targeting towards the end of this year to make those available.
Great. Thank you so much, and thank you, obviously, for all the work that you're putting in over the past 6 months and, in general. Thank you.
You're welcome.
Thank you. That was our last question. I will now turn the conference back over to our host, Irene Iyer. You may proceed, ma'am.
Thank you. This is Irene Iyer. We appreciate your participation and thoughtful questions. Today's presentation and transcript will be made available on the CDRH Learn webpage at www.fda.gov/training/CDRH Learn by Friday, October 9th. If you have additional questions about today's presentation, please email cdrh-EUA-templates@fda.hhs.gov. As always, we appreciate your feedback. Following the conclusion of today's presentation, please complete a short 13-question survey about your FDA CDRH virtual town hall experience. The survey can be found at www.fda.gov/CDRH webinar immediately following the conclusion of today's live discussion. Again, thank you for participating. This concludes today's discussion.
This does conclude today's conference call. We thank you all for participating. You may now disconnect and have a great rest of your day.